moved it in 84.
Welcome to Alan and Rodrigo, our first speaker.
Thanks very much, Jeff. Is the mic on? Can people hear me at the back? Great, excellent. You'll see from the title of my talk in the program, it's about big trees, but it's not going to be anywhere as elegant as the sort of stuff that Tandy was talking about on Monday. Really, I say here it's an idiosyncratic search for feasible solutions because what I'm interested in are the sorts of problems that particularly motivate my own research and the types of work that we encounter at the University of Auckland. I'm in the Bioinformatics Institute there. The types of problems that we encounter there. So really, it's quite an idiosyncratic search through the space of problems, looking for solutions. The talk will be structured in 3 parts. And as we go through the seminar, I will be less and less certain about anything. I hope to end up in a state of effective entropy, and that will be conducive for some good discussion. The first part really is work that Mike Steele and I have done on maximum likelihood supertrees. This work has been submitted and in fact is available on the archive if you are interested in having a look at the document. The next part of the work is concerned with what we can do when we have very large data sets and we are interested in how to derive some population genetic estimates from these very large data sets. So the second part is concerned with population genetics. And the third part is the part that I'm most concerned about as the director of the Bioinformatics Institute, because we know that in the next year or so we're going to be inundated with data from the new sequencing technologies that are already available and the genetic sequences are going to be coming, falling on our doorstep anytime. So the question is, how do we analyze this type of data? So let's start by thinking about supertrees. Now, what do we want to do when we construct a supertree? We all know what we want to do. What we want to do is we want to build a big tree of big N evolutionary units when all we have available are small subtrees of little n, 1, 2 to nk evolutionary units. Now we have topological information in the form of trees, single topologies. Maybe we have distributions of topologies for each of these data sets. Maybe we have splits or distributions of splits for each of these data sets. The topological information can come from different independent sources. And of course, we all know that supertrees need not be binary, as in fact the individual trees as well. Now, conceptually, I like to draw a distinction between the 2 ways that one can think about supertrees. We can think of supertrees as simply summaries of topological information. We have this information here, and we want to derive some summary statistic of this information. Alternatively, we can also think of supertrees as a phylogenetic estimate, a good phylogenetic estimate. Now, it should be apparent that it's not always the case that what is a summary is going to be, in fact, the best phylogenetic estimate of the data. So I want to make sure that we have in our minds this distinction between a supertree as a summary versus a supertree as an estimate of phylogeny. And this will become apparent why I want to make this distinction in due course. So I'm going to say that what we really want to do as biologists is to build a supertree that is an estimate of a phylogeny. And I want to be able to say that as an estimate of phylogeny, I have certain desirable properties. For instance, if I add more data, I want it to be able to get to the true tree. That's a nice property of a phylogenetic estimate. Okay, so I want to think about supertrees as estimators of phylogeny. Now, if I'm prepared to think in this way, then it's quite nice because I have a whole arsenal of tools available to me from the phylogenetic literature that I could conceivably import to the supertree literature or the supertree reconstruction problem. So what might these tools be? Well, we know how to build trees with distances, for instance, phylogenetic trees with distances. So I could take distance methods and see if these can be used to build supertrees. And people have done that. I could take parsimony, and I could use parsimony to inspire me to construct a supertree, some algorithm that constructs a supertree using parsimony. Of course, we know people have done that. I could take maximum likelihood or a Bayesian reconstruction method and say, how do I get a supertree? Now, in fact, these are less common, but have been referred to, particularly in that quite useful book edited by Olaf Beninda Emmons called Phylogenetic Supertrees. And maximum likelihood was alluded to by Cotton and Page, maximum likelihood idea. And Bayesian inference of supertrees was discussed by Fred Ronquist, John Hulsenbeck, and Britton. So here what we've done is we've taken techniques that have typically been used in phylogenetic estimation and brought them to bear on the problem of supertrees. I want to focus on one of these techniques, the idea of using maximum likelihood to infer our supertree. So let's think about the most naive likelihood method one could come up with. Now, if we're going to use maximum likelihood, what we want to be able to do is look at the space of all possible supertree topologies, and to each topology we want to assign a likelihood score. What might this score look like? Well, for each tree, I could survey all of the subtrees that I have, and if all of them are compatible, I'm going to give that supertree topology a score C, some non-zero value. If at least one of those subtrees is incompatible with my supertree, in other words, it doesn't fit on my supertree, I'm just going to say it's 0. The likelihood score is 0. All right? This is the most naive way of building a likelihood function for supertrees. All right? So in this case, you would see that if I were to do this, it doesn't allow any conflict of topologies to contribute to my supertree. And it would, in fact, be equivalent to things like min-cut supertree and the Bill algorithm, etc. But it's not a very reasonable way of thinking about it. Why? Because we do expect conflict amongst our subtrees. So how do we deal with this conflict? Well, let's look at another possibility. Suppose we have access to distributional data. Suppose for each subset, we're able to look at every topology for that subset, every topology, and assign a likelihood score to every topology. So for each subtree, we have all of the possible topologies and all the likelihood scores associated with those topologies. So here we have, this is the subtree for the nth topology, for the ith data set, and this is the supertree, the jth supertree topology. So now you can see what I'm going to do next. I'm going to define a likelihood function that is simply the product of the likelihoods of the individual data sets. This di is the individual data set. This h represents the likelihood. And how am I going to get that likelihood? I'm going to get that likelihood this way. I'm going to scan through all of the subtrees, all of the topologies in each of the sub-data sets. And if it's compatible, I'm going to give it a value of 1. I'm going to allow it to contribute to the sum. And if it's not compatible, I'm not going to allow it to contribute to that sum. All right? So here, what I'm doing is I'm looking at the likelihood scores at for each subset of data, the likelihood scores, and I'm asking, what is the topology that is compatible with my supertree topology that I'm examining right now? And what is the likelihood score of that? How do I add it up? And can it give me a total likelihood score? So, of course, here in this case, we're going to choose the supertree that maximizes that likelihood from that function. The nice thing about this is that for every topology, I have a non-zero likelihood score. For every supertree topology, I have a non-zero likelihood score. Now, of course, this is a theoretical talk. I'm not talking about the practical issue of enumerating all of those topologies and assigning the scores, etc. But here, so this is the theoretical flavor of what what could be done. It does not include any information when your topologies are in fact in conflict. Okay. So if for each data set, anytime there's a conflicting topology, I don't worry about that. I go to the next topology for that data set, and it's only the ones that are compatible that I sum the likelihood scores for. Now, this is in fact related to the Bayesian method by Ronquist et al., except that they then take it further and look at splits. They break down the problem to look at splits. Okay? So what is the problem with this L1 score? Well, first of all, are we going to have the distributions of the likelihood scores for each subset? Not likely. Are we going to have the split support? Possibly. Maybe we're going to have bootstrap values? Possibly. Are we going to have the original data sets for the original, for those subsets on which we're building our supertree? Well, if we do, you can reasonably ask the question, well, why not simply concatenate the data to build one big tree? And that's a fair enough question. And of course, then if you did that, different models can be applied to different regions of the data set, et cetera. So you gain a whole lot more information. So this L1 tree doesn't sound as though it'd be very feasible. So what we really want is a nice likelihood function that can allow us to assign these likelihood scores to supertrees without access to the original data, the original sequence data, the original protein data, the original morphological data, etc. And without access to the original likelihood scores for all of the profiles for each subset. So, in a sense, we want to define this error model that takes a subtree and compares it to a supertree and assigns a probability that that subtree is going to be topologically incongruent with the supertree. So we want to derive a probability that allows for this error, this topological conflict. And this is a nice, reasonable model that one could think of. All right? So for each subset, we're going to define this likelihood, and this likelihood is going to be defined as a simple exponential decay function decaying on the distance between the subtree and a pruned supertree. Now we have to specify a pruned supertree because we're comparing this subtree against the supertree. We're going to chop off the leaves of the supertree that are not in part of the subtree, and we're going to compare it. That's the distance measure we're comparing here. And it's an exponential decay function. All right? This term Ci simply serves as a normalizing constant to make sure the probability sum to 1. The B's are going to tell us how fast that decay occurs. A large B tells us that there's very little conflict that's going to be tolerated. A small b tells us that it's going to allow quite a bit of conflict. So, you can see if you're using that function that it becomes pretty straightforward that the likelihood is in fact going to be maximized when the sum of bi times the distances is minimized. Now a note here, and I know Mark Wilkinson will be talking about this a little bit later. This function, in fact, is exactly the same as the one derived by Cotton and Wilkinson in systematic biology this year, when the distance is equal to Robinson-Fools and all the BIs are equal. The BIs then just fall out as a constant. And also the ML supertree, in this case, technical thing, is a median tree. But this function is the interesting function, isn't it? As I said, it's equivalent to statements about how easily a data set can give conflicting topologies. We can assign different values of B to different data sets, depending on whether, for instance, we may have more or less complex models of evolution and whether we trust those models of evolution, whether we believe that secondary structure is somehow going to influence the way the topologies are built, whether or not there's low phylogenetic signal or too much saturation in the data, whether or not we think that selection may have acted. All of these kinds of things could influence how B is chosen. Now, I speak of choosing B, but of course, we don't choose B in any sense unless we can apply this to some kind of Bayesian method, perhaps, where we allow B to vary as a parameter that we can choose, that we can come to estimate the posterior probability of. So, how might we apply this? All right, so this graph is a little bit complicated. Let me take the time to explain it. Up the top here is a 5-taxon tree. 1, 2, 3, 4, 5. 3, 4, 5. 1, 2, yes, right. You will see that there are, in fact, for 5-taxon trees, there are 15 such trees. And if you count here, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Couldn't fit it on another column. 15 possible 5-taxon trees. All right? This is all the possible 5-taxon trees for taxa from 1 to 5. These are 3 subtrees, 4-taxon subtrees. Okay, so here's 1, 2, 3, 4, 1, 2, 1, 3, 2, 4. I better look here. And 2, 3, 4, 5. So now what I want to do is compare each of these subtrees to each of these possible 5-taxon trees, because ultimately I want to build a super tree of 5 taxa, right? Because I have 5 taxa here, I want to build a super tree of 5 taxa. These are all the possible super trees I could have. This distance here is the Robinson-Fools distance. So simply I look at this tree, I look at this tree, there's a distance of 0. Because if I prune away, 1, 2, 3, if I prune away 5, I get exactly that tree. This tree and this tree has a distance of 2, because if I prune away 5, I get 1 distance. All right? So I just sum up all of those distances. So here's the total sum of the distances from the subtree, 2, 4, 4, 4, 6, 4, etc. And I look for the super trees that minimize that distance. And in fact, looking here, it's this one, and it's this one. That's it. That's all the example is showing. So it's a very long explanation for a rather trivial example, but I hope it illustrates the point. So this part of the talk is Mike Steele's, in the sense that the mathematics of proving these various properties come directly from Mike's elegant mind. So the first thing is the statistical consistency of this model, this L2 model. It requires this thing called the covering property. And the covering property, if I can say it in kind of simple words without mucking up the actual technical definition, basically it means that if I have subsets, if I can specify all subsets of 3 for rooted trees or 4 for unrooted trees, and those subsets, as I keep adding subtrees, don't disappear from the site. They keep being represented as I keep adding those trees. They don't disappear from sight, so they have strictly positive support as the number of subsets increases. Then that's fine. It's statistically consistent. L2 is statistically consistent. So this is really interesting, isn't it? Because now we have a way of looking at the consistency of supertrees, whereas we never had this before. And in fact, if you were to think of supertrees in terms of summaries, what's the equivalent kind of criterion that we're thinking of? Whereas now when we're thinking about it in terms of phylogenies, we are measuring it against the truth, i.e. the true phylogeny. So it's quite a useful way of thinking about supertrees, isn't it? It also allows us to now look at other supertree methods and ask whether those methods are consistent. Now, we know that L2 trees are often going to be MRP trees. But, as Mike has proven, in fact, MRP trees can be inconsistent. There's going to be a value of B such that even if I built MRP consensus trees, I would have this probability that the super tree that I build as I added more data sets would be different from the true super tree. All right? So MRP under under some values of B is going to be inconsistent. Turns out MRP is consistent when all the subtrees are quartets. Now, there was a recent paper by Degnan and Rosenberg, which was very interesting because they were talking about gene trees. And what they said was that if I have any species tree with more than 4 taxa, it is possible that the most likely gene tree for any gene is in fact going to differ from T, from the true tree. And if I took a simple majority rule approach to building my species tree, just by counting the trees that were most frequently represented across the genes, I would get it wrong. Now, why would I get it wrong? Well, I'd get it wrong, first of all, possibly because of gene reconstruction, but even putting that aside, even if we had a very good method perfectly reconstructing the gene trees, incomplete lineage sorting is still going to have a problem, present a problem. In other words, I can have lineages sorting differently from than what one would expect with the species tree. But Mike has been able to show that the statistical consistency of ML supertrees extends to handle this as well. So the upshot is that we have now a nice method for which one can look at properties like statistical consistency and therefore assess how these methods perform in terms of recovering the true phylogenetic tree. But questions obviously remain, as one would expect. For instance, I've only been talking about distances and using as my canonical example Robinson-Fools distances. But you could imagine other distances here, could you not? NNI, TBR, SPR, how do these perform? David Bryant raised an interesting question when we showed him this manuscript. He said, do we even need a metric distance? Do we need the triangle inequality? Good question. We've used a simple exponential decay function, but what is an alternative good function that one could possibly use? And finally, how can we look at likelihood supertrees and extend them or show the congruence, as it were, with other supertree methods? So I think these are some of the questions that remain, and hopefully people here and once the paper comes out, we'll be able to extend the work. All right, switch tack totally. Let's talk about population genetics, the coalescent, and big trees. Now, not so many talks on the coalescent. Yesterday, Jotun gave a very nice talk on the coalescent. In population genetics, we have the standard Wright-Fisher model, the sort of paradigm of population modeling, population dynamics, where we have perhaps in the simplest case a constant population size, a constant sized population. It's pan-mictic. All individuals can interbreed with each other. Every individual in each generation has an equal propensity to produce offspring. And there are no selective forces shaping the population. And of course, after some finite interval of time, there's going to be the loss of lineages from the past to the present. There's going to be the loss of lineages just by chance alone, so that all of the lineages at some point in time derive from the same common ancestor. Now, John Kingman derived a continuous time approximation of the Wright-Fisher model with large N. He called this the coalescent, the N coalescent. It basically describes the genealogy of a small sample taken from this large population. The nice thing about this description of the genealogy is that it tells us about the expected times and the statistical distributions of coalescent intervals, the times of the coalescent events, and therefore of the coalescent intervals itself. And those times are related to the population size. So one can imagine that, for instance, when you have a very large population, that it takes longer for you to find a common ancestor with somebody else in the audience. But if there's a very small population, it will take you a lot quicker to find a common ancestor with somebody else, randomly selected from the population. The expected time for each coalescent event is interval is exponentially distributed, and this is the mean, 2n divided by n times n minus 1. Okay. Now, of course, we are dealing with sequence data here. I'm going to assume that we're dealing with sequence data. We don't measure time in real time. Instead, we measure time in substitution. So we rescale everything by substitutions. And so instead of talking about population size now, we talk about this joint parameter n mu, or 2n mu, and that's called theta. So because we rescale time, we now talk about generations in terms of theta. Everything else remains the same. So if I give you a genealogy where I specify the coalescent intervals, you can write down for me the probability of obtaining that genealogy given a value of theta. And that probability is simply the product of the exponential random variables scaled by theta. All right, and remember, time, once again, is measured in substitution units, and theta is 2n mu. So you are able to write down, as it were, the likelihood of theta for any given genealogy. And if you are able to write down the likelihood, you're able to solve that likelihood. And in fact, you're able to get theta, the maximum likelihood estimate of theta, which is simply the average estimate for each of these intervals here. And you can get the asymptotic variance for that likelihood estimate of theta as well. What if the genealogy is not known? So now what you have in your hands are the sequences. You have no genealogy. Well, you could do something like build a phylogenetic tree first and then pretend that you know that to be the genealogy and then go ahead and estimate theta. Sure, that's fine. Yes, you can do that. The problem with that, of course, is that we know when we build phylogenetic trees, there's uncertainty involved in that reconstruction. And so really what you want to do is you want to say, let's look at all possible phylogenies, all possible genealogies, and weight our likelihoods of theta from the genealogy, weight this by the likelihood of that genealogy itself. And this term here is simply the standard phylogenetic likelihood, the standard communal garden phylogenetic likelihood that you get from phylip or Pope star or phyamel, whatever. Okay? So all I'm going to do now is I'm going to sample every possible genealogy, and I'm going to calculate this, and I'm going to weight it by its likelihood. And I know that I can't do that. But before I tell you feasible ways of doing this, it's interesting to notice that when we're thinking of the coalescent, we can break this thing called the genealogy into two bits. The first bit is really the ordered history of the coalescent events. Now, what do I mean by ordered history? In this genealogy, the order of the coalescent events is one ordered history. So A, B coming first, and then C and D coalescing, and then C, D, E coalescing, and finally all of those coalescing, represent one ordered history. I could have another ordered history with exactly the same topology where I have CD coalescing first, and then AB, and then CDE. That's a second ordered history with the same topology. And of course, I can have the third ordered history, which is simply CD, then CDE, and then finally AB here, and then ABCD. So I have various ordered histories, and I have coalescent intervals. We can write the genealogy, therefore, as a kind of a combination of ordered histories. Any one genealogy is specified by the combination of the ordered history and the coalescent intervals themselves. The nice thing about this is that whereas I will have an infinite number of genealogies because the branch lengths vary, etc., in terms of ordered histories, I have a finite number of ordered histories that I can enumerate, right? And that's the value there. So now what I can do is take my function here, my likelihood function to get theta, and break it up into ordered histories and coalescent intervals. All right? So all I've done here is replace G with H and C. But here, something has changed. You'll see that theta only tells me about the coalescent intervals. Theta has no information about the precise nature of the ordered history itself. So here I have the coalescent intervals, and here I have the full genealogy D. Okay? Why is this useful? Well, it's useful when I have a very small subset of samples, of sequences. Because here I can enumerate all my ordered histories from 1 to 180. When there are 5 sequences, there are 180 ordered histories. And for each ordered history, I can simply adjust the coalescent intervals independently and sum over all of those. So I can get pretty nearly exact likelihood values for different values of theta. Right? Just simply by enumerating over all ordered histories and fiddling these 1, 2, 3, 4 intervals. Okay? I can do this with small numbers of sequences. Small n. Small n is possible to enumerate all ordered histories and compute the likelihood in a reasonable amount of time. With large n, we can't do that. So what do people do? Well, with large n, they use importance sampling, they use MCMC. And several people have done this. Most people have done this. Everyone has done this. Okay. So here's a question that comes from the mind of perhaps an applied biostatistician. I have this big data. Why can't I break up my data set into small bits and for each small bit get an estimate of theta and then somehow pool those estimates? But you have to ask the question, first of all, why do I have such a large amount of data? People have shown, for instance, that to get good inference on theta, you only need 40, 20, 30 sequences. So why am I dealing with large amounts of data? I'm dealing with large amounts of data because I'm dealing with biologists. If a biologist has in her hands 2 PCR primers that work and a 96 well plate, how many sequences will that biologist produce from the same population? At least 96. In fact, because I know my primers work and next day the PCR machine is free, I'm going to have 192 sequences, right? Because I know that it's easy to get these to work. Now, we know that with the coalescent, in fact, it's better to get multiple loci. But you know, each time you have to amplify another locus, you in fact have to optimize your PCR, etc. So it's a lot easier to get lots and lots of sequences from the same locus. So you're going to get a biologist coming to you with 150 sequences, with 200 sequences. Are you going to tell that biologist, I only really need 40? I don't need the rest of your sequences, because I don't think that'll go down very well. So what do? We do the things that we do with MCMC. We do the things that we do with importance sampling. The problems, of course, is that all of the surfaces that we're dealing with in terms of topologies, in terms of likelihood, in terms of these parameter spaces are all quite bumpy. And often we may have difficulty converging, or you have to run it over very long periods of time. Other problems as well. So here's the naive proposal. I've got a large set of sequences. And what I'm going to do is I'm going to break them up into small subsets. Small, easily manageable subsets. And for each of those subsets, I'm going to estimate theta. And I'm going to estimate an overall value of the mean simply by averaging over all of those theta. Sorry, that should be a 1 over n, where n is the number of subsets. I'm going to average over all of those. And I'm also going to take the variance of theta simply by just calculating the variance of my estimates. A totally naive approach. So how does that naive approach work? How does it perform? So I did some simulations, or rather my programmer did some simulations. Using SimCore, we did 20 data sets with 70 sequences, population with a constant n mu, K2P model of evolution, very preliminary stuff. So for each data set, we broke it up into non-overlapping subsets of 14 sequences. So there are 14 sequences for each of these 20 data sets. We obtained the maximum likelihood and Bayesian estimates using Lamarck and using mean of means and standard deviation of means, and using BEAST. Bayesian estimates using pooled median and pooled 95% highest posterior densities. All right, so let's have a look at how they perform. So here's these 20 pairs. The short bars, the small bars, represent the full data set in a single run. So we gave Lamarck the 70 sequences, and we ran those 70 sequences. And the long bars represent our estimates of the confidence interval and the means based on my naive view of splitting up the data. So one of the things I often tell my students when I teach them statistics is you only need as much certainty in your data as you need to reject a null hypothesis. So if your null hypothesis is 0.8, you're fine with this approach, aren't you? If your null hypothesis here is 0.4, you're not so good. All right? But still, you can see that it's not too bad. Some of these, at least, appear to be comparable. Some of these ideas of splitting up the data set appear to be comparable. Interestingly, if you look at the times, what have I done? My 14 subset data has gone down to 16 minutes a run, but my complete run takes an hour. Now, of course, I have to run 5 of those 14 data sets. So if I run them sequentially, that's not good because it's more than the time it takes to analyze my entire data set. But if I have a cluster, then it actually works out not too bad. It's 1/4. It's even more striking with BEAST, though, with the Bayesian analysis. So here, the Bayesian analysis, you see the same pattern. The full data sets have very tight confidence intervals. The big data sets don't have tight confidence intervals. I'm sorry, the subsets don't have tight confidence intervals. But look at the difference in runtime. That's 3.7 minutes per 14 sequence subset versus a complete run of 1.3 hours. That's a factor of 20. If I can distribute 5 of those, I'll be able to speed up my calculations 20 times. So this is why I want to do this. All right? The problem remains, of course, the variance. Why is this a problem? We know that these values of thetas that we get from each of these subsets are correlated. And they're not just correlated because there happens to be a true theta and each of these values are instances, sampling instances of that true theta. It's not just because of that. It's because there are these overlapping genealogies. So if you have a look, let's say I sample 2 sequences first. What is the expected time to the most recent common ancestor for 2 sequences from a haploid population? Well, from any population, theta over 2. 2 sequences. I've got this to one side. What is the expected time for now another 2 sequences that I've sampled from the population at a later point? Is it also going to be theta over 2? Well, no. It turns out that that expected time has the first time, first observed time in it. It has this first observed time in it. Why is that? Well, it's because at least some of the way that this pair of sequences can relate to this pair is so that C falls with A and D falls with B, which forces the root to be identical to this root here. So the root of here is forced to be identical to the root of here. A kind of coalescent entanglement phenomenon. Right? And so, in fact, if you have these correlations between apparently independent data sets, they're not independent. So how do we derive an estimate of theta? Okay, so finish this part of the talk. Biologists can and will collect a large number of sequences from a given locus. Can we increase computational efficiency by splitting up the data? And if so, how? And more importantly, what is the right way of calculating the variance of theta bar? Okay, next, new sequencing technologies. As I said, this is the part of the talk where the uncertainty really reaches its peak. So most of you should be aware of the new types of sequences that are out there. These are sequences that basically produce a large amount of very short fragments randomly from a genome or a collection of whatever DNA you have available. Millions of fragments between 50 nucleotides and 300 nucleotides, depending on which technology you're using, within 24 hours. So you can have 10 to the 6 to 10 to the 9 nucleotides in a single run within 24 hours. And people are already doing other technologies on this. And there's a whole lot of work being done on the computational analysis of this large amount of data. But they have tended to focus on, first, whether we can reconstruct the complete genome of whatever organism we've given to those sequences, de novo genome assembly, or whether we can put those fragments on a scaffold. What's the best way of doing that? How do we avoid repeats? How do we make error corrections, etc.? So they're focused on those types of problems, where really we're thinking about still, and the old idea of coming out with a sequence that reflects the entire genome in one long string. And so I want to raise what I think is a reasonably intriguing question, that is, can we get away without assembling the genome? Now, for a few years, people have been thinking about how we can do phylogenetics without using alignments. And there are reasons why we would want to do this. And in fact, Hurl, Rigoutzis, and Reagan reviewed, in an extensive review last year in Evolutionary Bioinformatics, they reviewed a whole series of alignment-free phylogenetic methods. And basically, these can be grouped into two types of methods. One is using word frequency spectra, and the other is using compression-type procedures. So just give you an idea, for those of you who may not be familiar with this, a word frequency spectrum, we simply define a word length, k. If this k is 3, for instance, then it's equivalent to codons. And what we do is for each set of DNA that we have, we scan the entire sequence and look for the frequencies of all possible k words. And then, for each genome, we have the spectrum here for the number of k words. So as I said, if it's for 3, you have 64. You have the spectrum of 64, the frequency spectrum, and you compare it against another genome. And by building this distance metric, you can use a distance metric. You can normalize according to word frequencies. You can change the type of distance you use. The earliest ones used Euclidean distances. You can change the way you calculate expected frequencies to normalize. Once you've got your distances, you build your phylogeny using neighbor joining, which seems to be the tool of choice for distance-based methods. So here's an example from Hall et al., just showing how some of these distances perform relative to the reference distance. So here's the reference distance on each of these graphs. Each of these graphs is the performance of different distance methods, different word spectrum distances, and here are the reference distances. And you can see that some of them aren't too bad, and some of them are absolutely abysmal, right? But the ones that aren't too bad really aren't too bad. Okay, so here we have a distance that is 2 substitutions per site, and you seem to be recovering that pretty well. So it seems to me that what we have with these short read sequences is a way of generating spectra, because each 15 nucleotide fragment is a word. And I have these words that I can align, and there are millions of them, and they constitute, to me anyway, a spectrum. So can I use these word-based alignment-free methods to compare these sequences? So what I'm saying here is, let's not worry about genome assembly. Let's try to compare these spectra. So that's one thing. That's one possibility. I don't know if it can be done. The next type of tool that I think is also very intriguing is this idea of using compression statistics. So compression features large in the computer science literature, of course, a lot of sophisticated math behind it, various ways of compressing things. But the idea is very nice, isn't it? If I want to know how similar two things are, I look at the amount of compressibility when I put them together relative to their compressibilities when they're apart. In other words, how much shared information is there in both of these data sets that allow me to compress them? Which is a really nice way of measuring the similarity between sets of large data, very large data sets. So Benny, I know, has got a paper doing some nice phylogenetic work on this. Can we use this to look at these large data sets of millions of sequences, short read sequences? No reason why they can't be used for single genomes, where each of your samples represents a single genome. But I'm more interested because I have a background in virus evolution, and here we have diverse communities of sequences. And if I have a diverse community of sequences, I can put these into my sequencer, get out these fragments, and I can compare, using this compression technology, two communities. So if I have two individuals infected with HIV, for instance, which is what I study, I can ask how similar these two are in terms of their virus population. I can ask how similar they are over time in terms of their virus populations. I can look at bacterial communities in different ecosystems, and I can ask, is this ecosystem have the same type of community in this ecosystem? So it seems to me that these two technologies can be used to summarize similarities between the data. So therefore, I would say that with these technologies, there is what I call a conceptual mapping between the actual data, the physical data that we get out of these things, and the types of methods that we've generated for alignment-free phylogenetics. And it may be profitable to explore how well word-based and compression-based phylogenetics apply to these data types. So I just want to finish off, of course, by thanking Mike Steele. Helen Shearman is my programmer, the organizers at the Isaac Newton Institute. And just to make two adverts, if I could. The first is that Olivier is organizing next year a conference, Mathematics and Informatics and Evolution in Phylogenetics. It's going to be near Montpellier from the 10th to the 12th of June. You're all welcome. And also just 2 job vacancies at the University of Auckland for evolutionary biology and applied mathematics. Thank you very much for your attention. Eric first. Then.
Did you ever think about doing a full model-based approach for your likelihood calculation? So instead of doing a distance, cutting out the
Yes, that's a very good question. Yes, the idea, we had thought of that. We wanted to try to bring out this framework first because, as you can see, there's all these nice material wrapped up around it with the consistency analyses, etc. But that is definitely a profitable way of doing, to look for the types of things that can cause conflict. I think that is an obvious next step. Daniel.
Yeah, so we've been looking at what's called metagenomics. So what you talked about here, where you sequence some anonymous DNA from a sample of water or something, and then what we do and what other people do is, along the lines of what you said, is you compare the reads that you get against reference sequences.
Yes.
And so what I think, what works for us is that we use BLAST against different databases, but we also tried, for example, that Tetra program that does tetranucleotides, and what we found is that the reads are just too short to use anything, to use any kind of signatures of this type. You just don't, if you only got 200 base pairs in a read or 100 base pairs, there's no way that you're going to be able to say, okay, this is from this organism or from that organism. So I think...
but that wasn't what I was thinking, was to take the entire set of 200 reads and put it and break it up. You know, use the entire set with without actually aligning it. Now, what are you going to miss? You're going to miss the joins, aren't you?
Well, you've got a metagenome. That's a whole lot of reads from many different organisms. So you might, you know, you've got a handful of soil.
Ah, right. I see. Sorry, I misunderstood. Yeah.
A billion different organisms in there. So you've got a couple of reads from each, and so you don't know which read. None of the reads overlap because you've got, even if you've got 10 million fragments, there's still not enough to actually be dense enough that anything overlaps. So you've just got these individual reads. You've only got 200 base pairs, and just by counting nucleotides and denucleotides or whatever, you're not going to see where that came from. But
are you thinking that the purpose there would be to identify the organisms themselves?
That's what we want to do in metagenomics, exactly. You've got a bucket, Craig Venter gets a bucket full of water.
Right. But I have an idea that what I want to do with the virus stuff, for instance, is not that, because I know it's HIV. What I want to do is I want to compare the communities genetically from one to the other. So I know I've got HIV here, I've got HIV here, but how similar are they?
But with the compression, would that...
I don't know.
That wouldn't tell you anything. I mean, biologists won't be able to interpret that. They say, okay, this data set gives me this number, this one gives me that number.
But if I put the two together, so if I have 5 patients, all right, and I have communities, and I have my HIV communities from 5, I compress, so I have 5 compressions, and then I do all pairwise compressions. So I pool them together and I compress, and I work out how much it's been compressed when they're together relative to when they're apart. That gives me a measure of similarity, doesn't it? Yeah, and so then in that case, I'm able to tell about I'm able to tell how genetically related communities are in the different hosts. Now, in the environmental one, I'm also thinking of this, because what we're doing, for instance, some of the work that we're doing involves wastewater treatment, where we want to know whether the community in a sewage pond, for instance, is at the point where it's going to become anaerobic, so it's going to become very smelly. Now, how do I test this? Well, I can take the genome, I can take the community from there, and I can compare it without having to go through and identify the organisms, actually compare it to all of the other timescales that I've had when the things have been fine. So in this respect, you're right, Craig Venter, the idea that most people want is to actually identify the organisms there. But I want to say, but then you see, what do people do after they've looked at the organisms there? They then make a secondary inference, and they say, well, this organism is there, therefore it means that the community has this kind of feature. Whereas what I want to say is, this is the genetics, the genetic material that is there, and this is the raw material that correlates with being in this type of environment. So, yeah.
I want to ask you about, you spoke about the genome spectrum, so we've been working with this, and first time, did you find that with genome spectrum, would you be able to really build phylogenetic trees? Because what we know is that, for instance, the genome signature, if you call it, varies along the genome.
Yeah. So various people have tried with the genomic spectra to build phylogenetic trees, haven't they? And they've had more or less success depending on how deep those trees are. So it seems that with reasonably, mid-range depth of trees, you can build these trees. And it makes sense because if you think about it, if I look just at codon frequencies of different organisms, I can detect phylogenetic structure just in codon frequencies itself. And codon frequencies are nothing but word spectra. Yeah?
This is quite restricted.
Yeah, it is. It's restricted. But if I can already detect phylogenetic similarities and structure within codon frequency tables, then I should be able to detect it in word spectra.
Any other questions? Okay, well, we'll take a break then. So could the next speaker, I think Professor Emerson, come and have a quick word from you. Thanks.